Hi, welcome to my video on Tetrahymena thermophila, a fascinating organism with 7 sexes and 2 nuclei. At first sight, Tetrahymena might seem just like any average microorganism, but despite this unassuming appearance, it has been the subject of much research all the way back to the 1950s, and much of this research has led to incredible revolutions in biology. For example, the Nobel Prize-winning discoveries of telomere structure by Elizabeth Blackburn and catalytic RNA by Thomas Cech were facilitated by studies on Tetrahymena. Tetrahymena is a unicellular eukaryote, which is part of a group of protists called ciliates. Here are a number of ciliates on the screen. Ciliates are characterized by the hair-like appendage that they have on their surface, which is called cilia, and you can see it swimming around with its cilia in this video. Most of the time, Tetrahymena reproduces asexually, but it can also reproduce sexually every so often, especially when it is starved, and this will become quite important later on. So far, nothing special, but here is where Tetrahymena starts becoming very strange. Tetrahymena, like all ciliates, has 2 nuclei in the same cell at the same time. The smaller nucleus is known as the micronucleus, MIC, and the larger one is known as the macronucleus, MAC. The micronucleus is diploid, and it contains 5 pairs of chromosomes. It behaves pretty much like you would expect it to. It undergoes mitosis during asexual reproduction, and it undergoes meiosis during sexual reproduction. Importantly, however, it is silent. None of its genes are transcribed. It merely sits there and stores information. The macronucleus is much weirder, and we'll get into the details later. For now, just think of it as a messy nucleus. Its genetic information is derived from the micronucleus, but it contains many copies of everything, with some parts missing and much smaller chromosomes. As we'll see later, it doesn't even divide by normal mitosis like the micronucleus does. Its role is to act as the site of transcription. This is where RNA is produced within the cell. When 2 Tetrahymena thermophila join to reproduce sexually, the micronuclei of both cells undergo meiosis. The macronuclei just sits there, and they are eventually destroyed. The details are not too important here, but essentially the 2 cells trade part of their micronuclear DNA, and they each end up with a new micronucleus. The new micronuclei now contain a haploid set of chromosomes, so 5 chromosomes, from the other cell, and another haploid set of chromosomes that they conserved from before. Essentially, half of their micronuclear DNA was swapped during this sexual reproduction. After a few more steps, which you can see in the diagram here if you want to pause the video, both cells divide in 2, and in the end you get 4 cells, each of which now has 2 identical nuclei. So let's take a look at one of these 4 cells. Initially, its 2 nuclei have the exact same DNA. They are both clones of one of the 2 micronuclei that was formed during sexual reproduction. However, as time passes, one of them is transforming into a macronucleus, while the other one stays as a micronucleus. Macronucleus formation is quite interesting. Firstly, the 5 pairs of chromosomes are fragmented into hundreds of smaller chromosomes. Some genes are also marked for removal and are not incorporated into the final macronuclear genome. Approximately 200 or so distinct mini-chromosomes, each containing completely different genes, are created in this process. Each mini-chromosome will be present in 2 slightly different versions with different alleles, which reflects the fact that the original micronucleus is diploid. After this fragmentation and modification process, the mini-chromosomes are all replicated multiple times within the macronucleus, which leads to the production of about 45 total copies of each mini-chromosome. For any given gene in the fully developed macronucleus, 22 might be of one allele, and 23 might be of the other allele, for example. Now, sexual reproduction does not actually happen very frequently. It only happens when the cells are starved and they are already sexually mature. Most of the time, Tetrahymena reproduces asexually. During asexual reproduction, after duplicating their DNA, of course, both the micronucleus and the macronucleus split in 2, and the cell simply divides into 2 daughter cells, each with a micro and a macronucleus. While the micronucleus goes through normal mitosis during asexual reproduction, the macronucleus cannot do so, as its short chromosomes do not contain centromeres, which are required in normal mitosis for spindle fibers to attach and segregate the chromosomes properly. Instead, after duplicating its DNA, like normal, in the S phase, the macronucleus divides in half without making sure that each side receives the same number of alleles for any given gene. The genome is not equally partitioned, and this is called amitosis. Now, when you think about it, amitosis has a pretty fascinating consequence. Because allele copies are distributed randomly between the two daughter macronuclei, certain lineage of descendants will, by chance, accumulate more copies of a certain allele than the other. An example of this is shown in the diagram here with a red and a green allele. As such, even if the macronucleus that was formed after sexual reproduction starts heterogeneous for a certain gene, some of its descendants will become homogeneous for one allele, and some will become homogeneous for the other allele, and this is called phenotypic assortment. Without even requiring sexual reproduction, amitosis is still able to create variety in clones, essentially, which is pretty stunning. This allows for short-term natural selection. Even if the macronuclear DNA is ultimately destroyed during the next round of sexual reproduction, this shortened variety, increases the chance that at least one of the lineages of clones will survive long enough to reproduce sexually again. So you can actually model this phenotypic assortment using a small program. Here's the output of a program that follows 100 hypothetical genes in the macronucleus as it undergoes many rounds of amitosis. On the left, you can see the number of amitosis. Each column represents one gene. Initially, about half of the 45 copies of each gene are of one recessive allele, and the other half of the dominant allele. After a few rounds of amitosis, phenotypic assortment starts to happen, and a green capital D is shown when all the 45 copies of the gene have become homogeneous for the dominant allele, while a orange capital R is shown when all 45 copies have become recessive. When the gene is still heterogeneous, a dot is just shown. As you can see in the simulation, after 25 generations, some of the genes started becoming homogeneous for a recessive allele, and this would almost certainly lead to observable changes in the phenotype of the cell. In this case, after 489 amitosis, phenotypic assortment is completed. The imaginary cell is now homogeneous for all of its 100 genes. I'll finish this video with another very cool fact about Tetrahymena that I hinted on earlier. It has 7 sexes, or 7 mating types. Each mating type is denoted by a different Roman numeral from 1 to 7. Now, how is a cell's mating type determined? Well, I won't get into too many details here, but essentially, every micronucleus actually has the genetic information to code for many different mating types. However, during macronuclear formation, after sexual reproduction, a random process will effectively choose one single mating type and get rid of all the DNA sequences that code for the other mating types. Once this mating type is fixed in the macronucleus, all of this cell's asexual progeny will also end up with the same sex. Since cells can only mate with cells that have a different mating type, this process effectively prevents inbreeding between fellow clones. Alright, I hope you enjoyed this video about Tetrahymena thermophila, an organism I found really fascinating. Sources are in the description if you are curious. And thanks for watching.